Okay, so ladies and gentlemen, good day all. So in this video, we are going to look at the concept of inborn error of amino acids metabolism. Introduction to inborn error. Inborn error of metabolism are genetic disorders caused by enzyme deficiency. So it is actually one of the diseases that can be inherited due to deficiency of one enzyme. The deficiency can be in the case of low concentration of the enzymes or absence of the enzymes completely in the body of the living organism. So when the enzymes concentration is low, that will lead to the inability of the body to produce enough concentration of the product by that enzymes. But if the enzymes is absent completely, it means that the product of that enzymes cannot be produced completely. So it is a disease that can be inherited. That is why it is called inborn errors of metabolism. These conditions affect the body's ability to metabolize amino acids properly, and early diagnosis and management are crucial to prevent severe complications. So as I said, as a result of the deficiency of the enzymes, it can actually affect the ability of the body to metabolize the amino acid properly. So you can see the definition of the disease. You can see that it is actually a genetic disorder that involves an enzyme deficiency. And this can actually result in the block or the enzymes can block or can lead to actually accumulation of this toxic substance or metabolites that is deficient synthesis of a product needed for the normal body function. So you see, listen, you have something like this. You have A as a substrate, which will be converted into E as a product. And you need an enzyme E1 to carry out this reaction. So if this enzyme is deficient, it means that A cannot be converted into D as a product. Therefore, the A will be accumulated and then the D will be absent completely in the body. And of course, this D may be actually a product that is essential or that is needed for the normal body function. So, but as a result of the deficiency of these enzymes, it means that the A cannot be converted into D. Therefore, A will be accumulated, and the accumulation of the A can lead to the accumulation or can lead to actually lead to toxicity, and that is why we call it accumulation of the toxic substance. So now, amino acids, we should know that they are the building blocks of proteins and they play essential roles in biological processes. So they are needed for protein synthesis. And of course, there are 20 amino acids that are found in proteins, and 9 of them are essential and must be obtained from the diets, while the 11 are non-essential amino acids. So non-essential amino acids are amino acids that can be synthesized by our body, while essential amino acids are those amino acids that we cannot synthesize them in our body. Amino acids are involved in neurotransmitter synthesis, hormones production, and energy metabolism. There are a lot of neurotransmitters like dopamine, serotonin, norepinephrine, epinephrine. They are all neurotransmitters that can be actually synthesized from amino acid, especially tyrosine. So this is actually how the amino acid looks like. Amino acid has a central carbon. This is a central carbon and it is called alpha carbon. And it has carboxylic group, amino group, and R side chain. And what will make one amino acid differ from one another is the R side chain. Like if the R side change is hydrogen, so the amino acid is glycine. If the R side change is CH3, it is called alanine. So each amino acid has its own side change. So each amino acid differs from one amino acid based on its side chain. So this is actually in the non-ionizable form of amino acids. Then the ionized form of amino acid, which is zeta ion, is when the amino group is positive and the carboxylic group is negative, but they still maintain zero charge. So the amino acid is important in producing some important hormones, energy metabolism, and the neurotransmitters. So you should understand, actually, ladies and gentlemen, if you ask to actually give 3 importance of amino acid or 4 importance of amino acid in the body, I think clearly you can mention it here. Number 1, it can be used for protein synthesis, it can be used for neurotransmitter synthesis, hormones production, and energy metabolism. So now, the inborn errors occur due to mutation in genes encoding enzymes responsible for amino acid metabolism. So mutation refers to a change in nucleotide sequence. A change in nucleotide sequence of a particular enzymes is what you call mutation. So when there is mutation, there is a likelihood there will be actually a deficiency of a particular protein or enzymes. It can actually, because deficiency or mutation is associated with so many things. Number 1, the presence of the mutation can change the structure of the enzymes, and that can affect the activity. Although the mutation can be positive and it can be negative, sometimes if there is a mutation, it can increase the activity of the enzymes. And in the other way, it can decrease the activity of the enzymes. In fact, it can even reach to a level where it will completely stop the activity of the enzymes. So mutation has these 3 consequences: to either increase, decrease, or block the activity of the enzymes. So then these mutations can lead to accumulation of toxic metabolites and depletion of essential compounds. Yeah, when we said accumulation, it means that the substrates will be accumulated because it cannot be converted into product, and then there will be depletion of essential compound. The essential compound here is the product, so it will be depleted because it's not longer produced in the body. So the resulting biochemical imbalance can cause serious clinical manifestations and organ dysfunction. Like, for example, look at this. This is where we have, you see, we have metabolite A, which will be converted into metabolite. But you see, as a result of the mutation in the DNA, it now actually produces the defective enzymes. So therefore, A cannot be converted into B. So as a result of that, the A will be accumulated. So accumulation of substance present in small amount. So therefore, this metabolite A will now be accumulated in the body. And then B cannot be produced. So if it cannot be produced, so it means that there will be a deficiency of the product. And also enzymes 2. Assuming A is converted into B, but if there is deficiency of enzymes 2, then B cannot be converted into C. So therefore, at the same time, B can be accumulated, and then there will be deficiency of metabolite C. So that is what we want to understand in the case of inborn error of metabolism. The inborn error of metabolism is associated with accumulation of the substrates and then depletion of the product due to mutation of the enzymes. So there are different types of inborn errors of metabolism of amino acids, and these include, the common types include urea cycle disorder, phenylketonuria, and branching amino acid disorders. Like, for example, let's say here, this is an amino acid here, which actually converted from it. There, you see, first you have the protein. So when you take the proteins, the protein can be digested to produce amino acid like phenylalanine. And then the enzymes here, phenylalanine hydroxylase, which is PAH, is now deficient. So if it is deficient, what will happen? It means that phenylalanine will not be converted into products. So as a result of that, there is an impaired amino acid metabolism enzymes, and that will lead to accumulation of phenylalanine. Then the product cannot be actually be produced, which is tyrosine. So that is the most important thing that you should understand. So this is actually in the case of phenylketonuria. You have the protein, you digest the protein, then it produces the individual amino acid, and one of the individual amino acid is phenylalanine, and then the phenylalanine cannot be converted into tyrosine due to the absence of phenylalanine hydroxylase. And that will lead to actually metabolic consequences. So, and each type of this inborn error of metabolism has clinical features, diagnostic method, and management strategies. So as we can see, we have different types of them, but you cannot actually apply the same clinical features. They are not going to have the same clinical features, but also the same method of diagnosis as well as the management strategies, because they are different. So phenylketonuria. Phenylketonuria is one of the types of inborn error of metabolism, and the disease is caused by the deficiency of the enzymes phenylalanine hydroxylase. So this is very important. So the key enzyme that is associated with phenylketonuria is phenylalanine hydroxylase, that is the deficient enzyme. So accumulation of phenylalanine can lead to intellectual disability and other neurological issues. You see, the major functions of this phenylalanine hydroxylase is to convert phenylalanine into tyrosine. And tyrosine is one of the important enzymes. So it's one of the important amino acid in biosynthesis of neurotransmitters such as serotonin, dopamine, norepinephrine. So therefore, as a result of the deficiency of this enzyme, means that those neurotransmitters cannot be produced. And as a result of that, it affects our brain. So that is why it is associated with intellectual disability and neurological issues. You see, this is what actually happens. A protein can be actually get from the dietary protein or endogenous protein. Of course, it will produce phenylalanine. And then the phenylalanine, in the presence of phenylalanine hydroxylase, it will be converted into tyrosine. But as a result of the deficiency of phenylalanine hydroxylase enzymes, the phenylalanine will be accumulated, and that will lead to the generation of phenylpyruvic acid, phenylactic acid, phenylacetic acid, and these are the biomolecules that actually accumulated and become toxic in the body. So in the absence of the enzyme, phenylactic acid and phenylacetic acid will be accumulated. So early dietary intervention can significantly improve the outcomes for individually phenylalanine ketonuria. So you know that since the enzymes is responsible for converting phenylalanine to tyrosine, so of course through dietary intervention, it means that since through these routes, the enzymes cannot actually produce tyrosine, then the tyrosine can be actually get from the diet. That is the dietary intervention. So that at least you have another source of getting tyrosine. So from there, it will continue to produce more of the neurotransmitters, which can improve the intellectual and neurological issues. So then the next one is maple syrup urine disease, which is the second type of inborn error of metabolism. Maple syrup urine disease is a disease that actually results from a deficiency in the branch chain alpha keto acid dehydrogenase complex. So this is actually the enzymes that is deficient in maple syrup urine disease. So these enzymes are actually enzymes that involve in metabolism branch chain amino acids. And this branch chain amino acid include leucine, isoleucine, and valine. So as a result of the deficiency of these alpha keto acids, the heterogeneous complex, it can cause neurological damage. This is actually the branch and amino acids, valine, leucine, and isoleucine. So a strict dietary restriction of these amino acids is essential for managing maple syrup urine disease. So it means that if you can now restrict yourself from taking diets that is rich in these branch and amino acid, it can actually lower the complications of the disease. So it means that if you have problem with this disease, you always look at the proteins that are less rich in the branch and amino acid that can actually have in managing the disease. So then the next thing is urea cycle disorders. Urea cycle disorders are caused by defect in the enzymes responsible for converting ammonia to urea. So elevated ammonia levels can cause severe neurological symptoms and can be life-threatening. So if there is deficiency of any enzymes of urea cycle, it means that the ammonia cannot be excreted to the urea cycle, and that will lead to actually elevation in ammonia levels, which can cause a serious disease. You see, this is a urea cycle. So if you look at it very clear, you'll understand that the first thing in this cycle is when carbon dioxide reacts with ammonium ion using an enzyme called carbamyl phosphate synthase 1, which actually leads to production of carbamyl phosphates. So the carbamyl phosphate contains with ornithine to produce citrulline using ornithine transcarbamylase. So ornithine transcarbamylase is an enzyme that condense carbamyl phosphate with ornithine to produce citrulline, and then citrulline with aspartate condense in the presence of arginosuccinate synthase to produce arginosuccinates, and then arginosuccinase enzymes hydrolyze arginosuccinate to produce fumarate and arginine. The arginine will be converted into ornithine with the help of an enzyme called arginase, which produce urea and ornithine, and then the urea will be excreted in the urine. So actually a problem with any of the enzymes from here can actually lead to accumulation of this ammonia, because it cannot actually be converted into urea, which is associated with neurological disorder, and it is also life-threatening. In fact, you can read on also what we call hyperammonemia for better understanding, to see how elevated level of ammonia can cause serious problem. So the treatment often involves dietary protein restrictions and administration of ammonia scavengers. So if you can do that, you can actually reduce the complication of the disease. And very importantly, the urea cycle is a cycle that takes place both in cytoplasm and the mitochondria. And as you can see, the condensation of or the formation of carbamoyl phosphates is taking place in the mitochondria. So, and of course, we have the rate limiting step. That is the step where you can regulate the pathway. You see where we have this term, that is carbamyl phosphate synthase 1. It means that it is the key enzyme. It is the regulatory enzymes in urea metabolism. So the next thing is tyrosine. Tyrosine is caused by the defects in the metabolism of amino acid tyrosine. So there are three types with varying severity and treatment approach. So there are three types of tyrosine. We have type 1, type 2, type 3. And the disease is caused as a result of deficiency of the enzymes that are involved in tyrosine metabolism. So look at it. The first thing now here is when we have an enzyme called we have phenylalanine. The phenylalanine will first be converted into tyrosine using an enzyme called phenylalanine hydroxylase. And then the tyrosine in the presence of tyrosinase will be converted into DOPA. The DOPA will be converted into dopamine. The dopamine will be converted into norepinephrine, and then the norepinephrine or noradrenaline will be converted into adrenaline. And this is actually taking place in the adrenal gland and nerve tissue. So it means that in the absence of the enzymes tyrosine hydroxylase, tyrosine cannot be converted into DOPA, and then the DOPA cannot be converted into dopamine, norepinephrine, and adrenaline. And of course, dopamine is a mood changer. Norepinephrine and adrenaline, they have a lot of physiological functions in the body in relation to intelligence, in relation to our heartbeats, and so many things. Then, of course, in the case of tyrosine, it can also be converted into DOPA using tyrosinase to produce, and of course, the DOPA can be converted into dopachrome, and then the dopachrome will be converted into melanins and the melanocytes. So it means that in the presence of the deficient, or in the absence of this tyrosinase, it means that the melanin cannot be produced. So ladies and gentlemen, from here I wanted you to understand, if you can get a compound that can inhibit tyrosinase, it can serve as a bleaching agent. Why? Because tyrosinase is the key enzyme that involved in the synthesis of melanins. And melanins is responsible for color of our skin. And the lower the melanin content, the fairer the skin it is. So it means that if you can get the enzymes inhibitor, that is the compound that can inhibit or decrease the activity of this tyrosinase, it means that you may actually produce the low concentration of the melanin, and that will actually increase in your fair skin color. And of course, at the same time, tyrosine can be converted into fumarate acetate, from fumarate acetate to fumarate, and then to glucose, and this usually takes place in the liver. The fumarate acetate can be converted into acetoacetate, which actually involved in the general catabolism. So this is actually what happened in the case of tyrosine. Like for example, type 1 tyrosine can lead to liver failure and requires a low tyrosine diet and medication. So this is actually, ladies and gentlemen, a brief on the tyrosine. So the next thing is actually argininemia. Argininemia is characterized by deficiency in the enzymes arginase. So the absence of arginase enzymes is associated with argininemia, and it leads to accumulation of arginine, and this can cause neurological impairment and developmental delays. So this is actually the major issues associated with arginase deficiency. You see, this is where we have a normal diet from protein from diets or protein from muscles will be digested to produce amino acids. Then the amino acids will be converted or will be hydrolyzed to produce ammonia. So in the actually absence of arginine enzymes, or arginase enzymes is responsible for producing urea in the urea cycle. That is one of the major enzymes in urea cycle, arginase, which actually converts, as you can see it from here. In the urea cycle, arginase is one of the enzymes that convert arginine to ornithine and urea. So if there is deficiency of the enzymes, as you can see from here, then the arginase enzymes cannot actually produce urea. So that will lead to accumulation of ammonia. So the ammonia will be built up, and that will lead to health problems. So arginine is one of the major enzymes that convert arginine into urea and ornithine. So the management include diet modification and symptomatic treatment of neurological disorders. So the next thing is cystinuria, which is cystinuria. Cystinuria is also a disorder affecting the transport of cysteine amino acid and other amino acid in the kidney. So you see, this is a cysteine. So if actually the cysteine cannot actually be transported from the urine to the blood, then it will now build off, and that can lead to the formation of kidney stone due to high levels of the cysteine in the urine. So increased hydration and medications to alkalize urine and help manage the condition. So you see, in the case of cystinuria, it is actually a situation or is a medical condition where cysteine amino acid in the urine cannot be transported back into the blood, and that is actually also associated with non-functioning of the of the transporters. So that is actually what leads to cystinuria. That is accumulation of cysteine in the urine due to the non-functioning of the transporters that carry them from the urine to the blood. So what are the diagnoses of inborn errors? So diagnosis typically involves biochemical analysis of blood and urine for abnormal metabolites. So you can actually take a blood or urine to assess, like in the case of cystinuria, you can take blood to check the amount or the concentration of the cysteine in the blood or urine. So genetic testing can confirm the specific enzyme deficiency or mutation responsible, because the best way, if you want to know whether the enzymes is deficient or not, is using the genetic testing. So this is actually how to do or how to carry out some tests that are associated with some of the deficiency of these biomolecules. If you can use blood, you can use urine. And from there, you will know whether the metabolites concentration is high or not. So newborn screening programs are crucial for early detection of certain inborn errors. So it is very important to diagnose the newborn. So then the clinical manifestation. The clinical presentation varies widely based on the specific disorder and the age of onset. So it is very important to know the age It's very important to know the to organize the disease at early stage. So symptoms might include failure to treat developmental delays and neurological disturbances, although each of these disorders have their own specific clinical presentation. Some disorders might present accurately while others might have chronic progressive symptoms. So this is actually how a lot of people are diagnosed with such kind of diseases. So now treatment efforts. So treatment often include dietary modification to restrict specific amino acids. So first, you need to understand, is it that the individual it is actually deficient in, let's say, tyrosine? Then you supply the individual with tyrosine. Maybe phenylalanine hydroxylase enzymes is deficient. So therefore, phenylalanine cannot be converted into tyrosine. So the best way, since the tyrosine cannot be produced from there, then you now get yourself to be taking foods that are highly rich in tyrosine, so that you actually help to manage the disease. And also supplementation with essential nutrient and essential amino and the amino acid might be necessary for growth and development. So in some cases, enzyme replacement therapy or CRISPR transplantation or liver transplantation might be considered. So sometimes if the enzymes is in the liver and we realize that the liver cannot produce such enzymes, you can actually replace the liver. You can do transplantation. You can do enzyme replacement therapy. You can get the enzymes maybe industrially and then you inject it. And another way, because there is this mutation, then you can use CRISPR technology to edit and correct the mutation in the enzymes. Or you can use gene therapy to remove the genes completely and replace it with the normal one. So you see dietary supplementation, dietary modification, enzyme replacement therapy, CRISPR technology, and gene therapy are the best way to tackle or to solve the problems of this hormone error of metabolism. So this is actually sometimes the enzyme deficiency might not always be as a result of mutation, even as a result of the post-transcriptional modification of the protein. So sometimes the enzymes can be produced, but it may have problems with post-translational modification. And this post-translational modification can be hydroxylation, phosphorylation, glycosylation, ubiquitination, sunulation, disulfide bond acetylation, and lipidation. All these are the example of post-translational modification of proteins. That their problem can also lead to the inability of the enzymes to carry out the enzymes activity. So monitoring and follow-up. Regular monitoring of amino acid levels and metabolic status is essential for effective management. So ongoing assessment can help adjust dietary restriction and supplementation as needed. So it is actually very important to be monitoring and following of the amino acid levels and the metabolic status, and that will actually help in the effective management and the treatment of the disease. Multidisciplinary teams, including dietitians, geneticists, and metabolic specialists, are vital for care, especially in this kind of situation. You need nutritionists, you need geneticists, and you need metabolic specialists. So then the next thing, of course, is the impact on quality of life. So inborn errors of metabolism, inborn errors of amino acid metabolism can significantly affect the individual's quality of life. Because if you have a problem with any of that disease, it can affect your health status. So early intervention and proper management can improve the outcomes and enhance life quality. Psychological support and education for families are also important components of care. So ladies and gentlemen, this is where we are going to stop. And I hope you will find this video very interesting, and please don't forget to subscribe and join the YouTube community. Bye bye, and see you next in my next video.